So next, we have actually 3 speakers, going to be short talks, and they are young scientists who will share their reflection on David's influence on their scientific journey. So we'll start with Tamsin Samuels. So Tamsin was a PhD student with Ilan Davis in Oxford, not so long ago, I get. And she did work and overlap quite a bit with David when David was very busy with, I guess, single molecule fish in Ilan's lab. So Tamsin.
Hi, thank you very much for the opportunity to say a few words here. So yeah, I'm Tamsin. I was a PhD student and postdoc with Ilan Davis between 2014 and 2019 at the University of Oxford. I actually joined Ilan's lab directly from doing my master's project with Simon Bullock. So I think in both of those degrees, I'm sure I was absorbing a lot of David's mentorship indirectly through them. But once I joined Ilan's lab, this was, of course, during that era where David was in our lab for a couple of days a week, and he was coming to lab meetings and coming to seminars, and of course was also around to talk about science, as well as to tell us where to get the best asparagus or the best coffee cups in Oxford. So I'm going to start out just by talking a little bit about one of the projects that we worked on together, but super briefly. And so this was the main thing I worked on during my PhD, and this was looking at this observation that 2 RNA binding proteins, imp and syncrip, were regulating the size of the Drosophila larval brain. So I have a Drosophila larva on the left-hand side, and these are 3 images of the larval brains with the wild type in the middle. And when we would knock down imp, the brain would become a lot smaller. If we knock down syncrip, the brain would become much larger. And following up on this, we found that imp and syncrip were actually regulating each other, and the levels of these proteins was changing during the larval development. So for the first part of my PhD project, I did some genome-wide RIP-seq experiments where I was finding the transcripts that were bound by both imp and syncrip. And then I ended up following up specifically on the MYC transcript, which was bound by imp, because we knew that this had a well-established role in cell growth and proliferation. And so then I was doing some of these ISH experiments, or the SM fish specifically, looking at the MYC transcripts in the Drosophila neural stem cells, or neuroblasts. And this method allowed me to sort of count the number of MYC transcripts in the neuroblasts, but also measuring the intensity of the transcription focus. I could measure the rate of transcription of MYC, as well as its half-life and antibody staining to find MYC protein. And so I was then making all these measurements and comparing between both different types of neuroblasts, but also neuroblasts during larval development. And of course, this ended up with a whole load of data, and where I think my discussions with David were really important was actually pulling this together into a model. And he really pushed me to look beyond my specific experiments and the data that I'd gathered, but to think about this in a bigger context. And so we ended up with this model where the rate of transcription of the mRNA seemed to be regulated at a systemic and extrinsic level, whereas then each individual neuroblast, depending on its levels of the RNA binding proteins, syncrip and imp, had this individual and intrinsic control, where imp binding to the MYC mRNA regulated its stability and therefore the amount of MYC protein, leading to this change in the cell growth and proliferation. And this ended up as a paper with David as a co-author. And especially during this project, I really appreciated how David could push me to think again and again about why this was important, but without making me feel just really bad about any of my data. So after I finished my sort of short postdoc with Ilan after my PhD, I joined Philippe Caram-Teixeira's group at the University of Cambridge and did a postdoc for 4 years there. And during that time, I was working then on the germline, but I stayed in touch with David, and he very kindly read a couple of my fellowship applications and gave similar long batches of really useful feedback. Again, one of the things I specifically remember about that was encouraging me to think most about the biological questions and what was the most interesting thing to do, and not just being centered on what I could do or what I thought I could do. Also during this time, I remember we both attended the 2022 BSCB conference in Warwick, and it really stands out in my memory how welcoming David was to introduce me to lots of other people that he knew. And that didn't extend just to me, it also extended to a couple of the other PhD students from Philippe's group who were there with me, who we'd never met before. And again, he was really introducing them to the community, and I think that meant a lot to them and to me. At the end of last year, I left academia and I'm now working at Storm Therapeutics, where we're doing cancer drug discovery, focusing on targets in the class of RNA modifying enzymes. But I wanted to end just with sort of a few pieces of wisdom from David that I still think about today all the time. The first one is this idea to keep an eye on the bigger picture, not get too focused on the details. To be precise with language, both in writing and in presentations, and the idea that that helps with clear thinking and also clear discussions and good scientific discussions. And the last one was no double tentatives. And so if you never heard this one, it was if you say, my hypothesis is that this might be because, and David would always highlight those and get rid of them on my writing. And I've now passed that on to many other students who I have worked with. So that's everything I wanted to say. I really got a lot from working with David, and I appreciate that I got to share some of that with you all today.
Very good advice here. Thank you, Tamsin.